Good morning, everyone. Let's start by taking up the application questions from lecture one. So these are questions that are found at the end of the study guide. I hope you had a chance to try them out. As I said, um, these will also show up on your first quiz, as a multiple choice question, not necessarily written. Okay, let's do the first one here. Let me read it to you first. Congestive heart failure, CHF, occurs when the ventricles fail to empty its content completely. Speculate what happens to someone if CHF occurs on the right side of the heart as well as the left side of the heart. So let's take a look at the heart once again. And we're going to have our four chambers. We have the right atrium here, right ventricle, and then left atrium and left ventricle. So let's take a look at what happens at on the right side with right congestive heart failure. Okay. So as the question described to you, in congestive heart failure, the heart is not emptying its content, meaning the blood is not fully being sent forward to the next part of the cycle. And so what ends up happening is that blood is going to start to pool here in the right ventricle. Instead of being sent off through the pulmonary artery completely, there are some leftover blood. And that could happen maybe because the muscle is getting weaker from various reasons, okay? Recovery from heart attack, or it could even be congenital congestive heart failure, meaning the person is born with this kind of a defect, right? So like if you're not able to empty the right ventricle, then the blood starts to pool in the right atrium as well, because it's just not enough space to fill up to completely empty the right atrium, right? Think of a car accident, right? Is once there's a car accident on the road, everything starts to build up and create a backlog, right? You're creating a traffic jam basically. And so, if we think about the vessels that are supplying the blood to the right atrium, which is, of course, the superior vena cava, as well as the inferior vena cava, now the right atrium is being blocked by these blood buildup, then you're going to have trouble emptying from the superior and inferior vena cava as well. So all this blood that are trying to return to the heart, they are having a difficult time doing that. And so it's not so much of a problem for the superior vena cava because gravity is going to help return them to the heart. But for the lower portion of the body, it's really gonna be problematic. Okay? So the blood starts to pool, pools in the lower portion of the body. Okay, and that basically increases the blood pressure in the lower portion of the body. And if you remember our discussion on osmotic pressure and blood pressure, this sudden increase in blood pressure is actually going to help push the fluid out of the capillaries more so than it normally would. And so these fluid that's going to be pushing out is basically going to result in swelling, which is also called edema of lower extremities. Okay, you're gonna have swelling in the legs, basically, in the foot. So that's what happens on the if the congestive heart failure happens on the right side. Now, on the left side, though, if it's happening on the left side, let me just draw this heart one more time. Left congestive heart failure. There we go. Gonna have the left atrium and left ventricle, and this time the blood is not emptying on the left side. It's not all of it is gonna be able to go through the aorta. You're gonna have some left over, and so the buildup is going to create, once again, a backlog in the left atrium. And the blood vessels that are supplying the left atrium from both sides, of course, they are going to have a hard time emptying as well. So these are pulmonary veins. And this time, instead of having accumulation of blood in the lower body, now we're having accumulation of blood in the lungs. Okay, so blood pools in the lungs. All right, there's going to be fluid buildup in the lungs. So we call that pulmonary edema. Okay, swelling, basically of tissue in the lung because of a buildup of fluid. And all that extra fluid can, increase the risk of getting infection, pneumonia, that kind of stuff. Right? So that's right congestive heart failure and left congestive heart failure. Um, yeah, and so that's like an application question. It requires you to think. I mean, I don't expect you to be able to come up with these terms necessarily, but you should be able to deduce the fact that if blood starts to build out, then eventually it's gonna, create a backlog. Does anybody have any question about that? Okay, number two, a heart block occurs when there is an interruption in conduction system of the heart. So if you remember, the conduction system is SA node, which creates the electrical signal that gets spread to the AV node, and then it goes to the AV bundle, and then the Purkinje fiber, right? And that basically is going to create the contraction, the cardiac cycle. So a heart block is basically an interruption along this path. In this case, it says the first degree heart block, the conduction from SA node to AV node is delayed. Right? So going as the signal spreads from SA to AV, it's being delayed. There is going to be a longer period of time, basically. What changes would be expected on the ECG for someone with a first degree heart block? So if you remember the ECG, I would just do it over here. Maybe I can grab an ECG diagram from the study guide. I think I included one. There it is. Okay, so here is the regular ECG. If you need a refresher, the P peak here corresponds to atrial contraction, and the R peak corresponds to um, ventricular contraction. And this here, this portion, oops, that's showing up. This portion is basically the signal, right? Signal going from SA node to AV node. So for someone experiencing a first degree heart block, because there's a delay, you would expect the distance, right, between P and Q to be extended. That's all. So that one is pretty straightforward, I think. All right. We are good with lecture one. Remember, your quiz is going to be next Monday. Please take the time to review the self quiz and everything. And also, I want to show you this. This is new. Do you guys get notification when I post new things on the course? Is it like a yeah? Okay, great. I'm not familiar with the whole thing yet. Now, yesterday I posted a bunch of stuff. If you go to the course homepage and you just scroll down to lab number one, um, so there is the PowerPoint that you can download and start working. But I created this lab video folder, okay, and I really want you to check it out. Me and this other faculty, his name is Temjeet. We film a bunch of stuff yesterday and then we posted online. And so some of them are things that you already learned, and it will be helpful for you to review them. So if you click on that, it'll take you to a YouTube video, and you might or might not have to watch a commercial. Oh, no, you don't. Okay. And then what happened here is I basically go through the heart model, and I guess it's like taking too long to load. But anyway, you can watch that later, okay? So basically it's the model that you'll be using in the lab, and then it's me explaining various features. And then Temjit also explain other stuff like blood circulation, so on and so forth. So this is definitely something that you should watch, not just for the lab, but as part of reviewing for your quiz as well, right? Today we're going to learn about blood type, and that can be confusing for some people. So we made a video on that later. All these things are related to the lab, but it's also related to what you're learning, right? So use those as studying resources uh, as well. Okay, so that's under the lab one folder, lab video. Um, Let me just reiterate the point a little bit. If you click on the lab package, you'll see that's the heart model that was in the video, right? So you definitely want to watch that first before attempting this lab. You don't have to hand this in, remember? Right? You just have to do it, keep a good copy yourself, and then you will do the lab quiz afterwards. Is that okay? All right. I'm a little bit out of it today because I'm not feeling too well. So just so you know, you don't feel like I am not 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 wanting to be here or something. It's not that. It's just that I am a little bit low on energy. But that's all right. Let me start the PowerPoint here. There we go. Hey, so lecture two. Lecture two is blood and lymphatic system. This is not on your first quiz, okay? First quiz is just all of lecture one. Blood and lymphatic system are related. Okay? The lymphatic system is basically something that is related to immunity, as you will see. So a lot of what happens in your immune system exists within the blood. So we'll do the blood first, and then towards the end of the lecture, we'll do a little bit of lymphatic system, which would be a nice connection between second lecture and the third lecture. So these are your blood cells. You have a lot of red blood cells here, and every now and then you would have a white blood cell. The white ones are more spherical compared to the red ones, and they are not a majority of the blood. Okay, only about 1% of your cell is white cell. So how much blood do you have? Approximately 5 liters of blood for someone who is around 70 kilogram. Of course, the bigger you are, the more blood you will have. And blood, as you know, is essential for life. We can, bleed a little bit and still be able to survive. You can donate blood, for example, take out a significant volume of your blood, and you'll be okay. But if you lose too much of the stuff, then we would definitely die. So what is it that's in blood that makes it so important? Well, blood can be described as a protein-rich fluid with cellular components. Okay, so the fluid portion of the blood is primarily made up of water. And in that water, there are a bunch of proteins, amongst other things, dissolved in it. Okay, these proteins have various functions. There are different kinds of proteins that you will find in this portion of the blood, and we'll be learning some of the key ones. So that fluid portion is what we call the plasma, and that makes up about 55% of your blood. The cellular components are collectively known as formed elements. That makes up 45% of the whole blood. So you probably know some of the formed elements already. I think I told you that from last semester at the end. There are red blood cells, there are white blood cells, and then there are one more kind of um of cells there. Does anybody remember what they're called? They help you stop the bleeding. Hmm That's correct, Cheyenne. That is platelets. Okay. So today we will be learning more about each of these cell types. It turns out there are various kinds of white cells, and they all have different functions, right? And that would take us through the first half of the lesson. When you take blood from a person, right, you can actually separate the blood using what's called a centrifuge. A centrifuge is basically a machine that spin contents at really, really high speed, and it will separate contents by weight. Right? I think we talked about this, right? Last semester when we were learning about um, the Hershey and Chase experiment, right? They were spinning things down, and then the heavier bacteria goes to the bottom, and the lighter viruses goes on top. This is similar. Heavier components of the blood will sink to the bottom, and lighter stuff will go to the top. And if you do this, the top portion is always going to be your plasma. And in a normal, healthy individual, the plasma should be a clear yellowish fluid. Okay, it's very similar to the color of apple juice, if you would. Okay, maybe a little bit darker, and that is mostly water with a lot of proteins in it. At the bottom, you would have the red cell. Red cells are also called erythrocytes. More on that later. And that makes up, the part of the form elements. But form elements also include the white cells and the platelets. And you can see because the white cells and the platelets are lighter compared to the red cells, they would be floating between the plasma layer and the red cell. And it's a very, very thin layer. It only makes up less than 1% of all your blood. Okay. But collectively, all these things are formed elements. This is what it looks like. This one, that's the only picture I can find online. It's not as clear as it should be. I'm not sure why. But typically, again, it's you should be able to see through it quite well. Okay, like apple juice, exactly like apple juice. I don't know why this one is like cloudy like that. And then what happens is they can freeze the blood cells, the red blood cells, and then they can freeze the plasma, right? And then later on, these can be used for transfusion, right? A person who lost a lot of blood, they need transfusion, for example. We're gonna learn about transfusion later on, I think today. It's important for you to understand the transfusion is not take a whole bag of blood and then just give it to someone. Okay, like that it doesn't work like that. Typically, they separate the blood into plasma and then they separate into red blood cells. You can see here it says red blood cells. Okay, and then it's AB positive. The rules for AB positive transfusion are the rule for red cell transfusion are different from the rules for plasma transfusion. Okay. And so, it's not like the movies where you can, you see someone hooks up a line and then directly give blood to another person. Even if your blood is complete match, like if you're B positive and the other person is also B positive, even it's like that, it's not very safe, okay? Because there are white cells in the body that, can start to react. All right, so we're gonna learn about the transfusion rules later. Uh, Plasma can be stored for, very long time. Talk about months, okay? Whereas red cells, they are more delicate. Even if you put them in the fridge, we're talking about weeks as opposed to months. Okay, let's take a closer look at the plasma. You can see here is fresh frozen plasma, right? You can freeze them because it's mostly water, right? It's fine. And then it says thaw plasma, um, and then you're supposed to put a date on it. Once you thaw it, you cannot freeze it. It's like ice cream, okay? You take it out, it melts. You can't refreeze them. It's gonna turn bad. So very similar to the plasma here. Like I said, it's mostly water. There are some dissolved ions in it, and it's all the ions that you can think of, okay? Like there is sodium, chlorine, right? There may be magnesium, right? A bunch of stuff, okay? Potassium. These are essential electrolytes that are needed in the body, and they are found in the plasma portion of the blood, okay? Nutrients like your glucose, your amino acid, everything that you need for the cells, they are carried in the plasma portion of the blood. There will be some waste product as well, right? Like carbon dioxide, urea. Okay? Of course, the gas is not going to be there once you separate the blood. It's going to leave. But like within the body, though, you will have the carbon dioxide in the plasma portion, and you will have the oxygen in the plasma portion as well. Okay, urea is a waste that you get when you metabolize proteins in the body. And most importantly, there are a bunch of plasma proteins. Okay, talking about hundreds of them, each playing an important role um, in keeping us functioning. All right, so this is the blood plasma. For people who donate blood, uh, they could choose to donate whole blood, so that's just taking the blood out of the body as is. But some people might choose to donate plasma only. Okay, and then so the blood comes out, it gets separated immediately, and then the cells goes back to your body, but the plasma is going to be collected. So plasma donation can happen more frequently. Whole blood donation, you have to wait about 3 months before you go back. All right. Let's take a look at the formed elements. Remember, the plasma makes up approximately 55% of your blood, whereas the formed elements, which are basically just the cellular components, makes up about 45% of your blood. Okay? So that includes red cells, white cells, and platelets. Now, the red cells is the most common type of cells in the blood, and the big portion that they make up at the bottom right here, that is called the hematocrit. Hematocrit. Right? It's a little bit confusing here. So let me go back to this slide to show you. You see the formed element is everything here, including the white cells and the platelets. If we are just talking about the red cells, it actually forms a little palette at the bottom. They're all compacted together. So that is called a hematocrit. Hematocrit. Okay, only the red cell portion. And this little part here, that's the hematocrit. All right. White cells help defend the body against pathogens, and you can see there are different kinds of white cells here. Compared to the red cell, is very scarce. Okay, not a lot of it. Red cells, we're talking about 5 to 6 million microliter. So 5 to 6 million red cells per microliter of blood, and whereas here we're talking about 5,000 to 10,000 white cells. Okay, it could change. It depends if your body is fighting an infection or not. These things can go up or down, but typically it's just 1% of your whole blood. Okay? And then platelets, we have more platelets than white cells. They are always there just in case you have cut and you need to stop the bleeding. Let's take a closer look at the plasma and the kind of proteins we have in it. It turns out the most abundant plasma protein, the most common plasma protein, is something called albumin. Okay, albumin. You can find albumin in egg whites, for example, okay? But your body makes it in the liver. And the main function of albumin is to maintain osmotic pressure. To maintain osmotic pressure. Now, you might remember last class. I hope you remember from last class. blood pressure is always pushing fluid out, right? So that's blood pressure. And to counter that effect, you need to have the osmotic pressure to bring fluid back in. And we talk about how the osmotic pressure is caused by a difference in solute concentration in the blood versus the outside. So the main contributing factor, the main contributing solute, if you would, in the blood, one of them anyways, would be the albumin, okay, along with the formed elements. Okay? So these albumin cause the concentration of the blood to be higher compared to the outside, and then it sucks the fluid into the capillaries. Okay, the albumin themselves are too big to leave the capillaries, so they will stay there inside. Right? And that creates the difference in solute concentration that will draw the fluid into the capillaries. Okay, so people who uh, drink like protein shakes after workout, much of it is basically albumin. Okay, it's protein. Now, if you look at kids who are malnutritioned, and this is something that you often see, when they promote World Vision or something, asking you to donate to help these kids. This is a very typical picture that they show with the hungry and malnutrition kids. They have a big belly. Okay, why is that the case? Okay. Well, when they are malnutritioned, they are not getting enough proteins in their diet. That's often the case. And when that happens, then the albumin level basically goes down. Okay, it goes down. And so the purple arrows are now going to be not as strong as before. You're still bringing fluid in, but you're not going to be able to retain as much of it because now the concentration difference is not as drastic, right? And so all that fluid that's being pushed out by the blood pressure will now stay in the tissue. Okay? And that causes accumulation of fluid and it results in swelling or edema. Okay? That's why you see these bulgy bellies. But you might wonder, how come it's only the belly? How come the hands and the feet don't swell up? Well, that's because when you move around, the muscle contraction, remember they kind of serve as a type of pump, and that pumping action, the contraction of the muscle, that actually helps return some of these fluid back into the blood vessels. Okay, but not so much the abdomen. When we move around, we're not exactly like doing sit up all the time, right? And so there's not as um, as much contraction going on there that will help return the fluid. And as a result, they create the swelling, swollen bellies. Does anybody have any question? Okay. There are other kinds of plasma proteins. Some are important in immune response. Others are important in coagulation. Okay, coagulation is just a fancy word for blood clot. I will introduce these to you first, and then we'll come back to it at a later point. For example, for the blood to clot, if there's a cut or something, you need these plasma protein. One of them is called fibrinogen. The other is called prothrombin. Okay, I'm just showing you right now. We'll talk more about them later on. These are made in the liver. Antibodies. Okay, antibodies are necessary for immune defense. Antibodies are the ultimate defense weapon your body is capable of making. If the antibodies can't help you fight off the infection, right, then there is nothing much your body can do. Then you need basically medication to help you get better. Right? The ultimate goal of your immune system is to create antibodies to fight off an invader. Okay, whether it's a bacteria or virus, you need to make antibodies. And these antibodies, once you make them, they will be in your plasma. Okay, plasma is the liquid portion of your blood. Okay, so albumin, fibrinogen, prothrombin, antibodies, these are just a few examples of all the proteins that are found in your plasma. Remember, there are a lot of proteins in your plasma, hundreds. So let's shift gear and talk about the blood cells. Where do they come from? The white cells, the red cells, and the platelets. It turns out they all came from stem cells that are found in your bone marrow. Okay. We talked about stem cells last semester at the end. Stem cells are cells that can become different kinds of cells in the body. But once you are grown up, then the stem cells are more restricted to a particular category. So in this case, when we are talking about the stem cells in the bone marrow, they can only specialize and become blood cells. Okay, they're not going to become like a lung cell or something. They have to stay within this category, right? And so under the right condition, some will become the red cell, some will become platelets, and then some will become white cells. And within the white cell category, there are many choices it could become, right? So you don't have to memorize these things. Okay, I'm just showing you. The process of making new blood cells, it's called hematopoiesis. Okay, this is the formation of new blood cells, not just red ones, right? All of them. Okay? It's called hematopoiesis. Hematopoiesis. So as an adult, the active bone marrow size is going to be reduced compared to when you're younger. For example, we have the femur. The femur is like your thigh bone. There are some active bone marrow there that makes new red cells. Sorry, makes new blood cells. There are some around your pelvic region. Um, the bone marrow there are also active and they could also make new blood cells, but not all the bone marrows throughout the entire body is capable of this process. Okay, only selected sites will be able to perform hematopoiesis. All right. You guys know bone marrows, right? It's the stuff inside the bone, basically, okay, in the core of the bone. Uh, this is actually my blood. Okay. One time, I was having a uh this episode of hives. Okay, I don't know why, but it's just a lot of hives, and then it's really itchy. And my wife, she studied bloods. Okay, like she works at the hematology department at the hospital, so she knows a lot about blood. And so she's like, why don't I take a look at your blood? Right? This was like during the COVID period, so like it was difficult to go see a doctor, I suppose. So anyway, she took my blood and then she examined it, and she's like, okay, these are the red cells, and then you have the white cells, and over here, yeah, the bluish stuff are the white cells. Okay. By the way, this slide here, it has been stained with some dyes. Okay. When people look at tissue, they have to stain it. Otherwise, everything is like colorless, and you can't really tell what's what. Okay, so these are red cells, the pink ones, and then the purple ones you can see it's not very common, right? Those are my white cells. And if you squint a little bit, you see these little purple dots, very small. Those are the platelets. Okay, those are the platelets. Anyway, so she looked at my cells, and then she found that I have a particular kind of white cells was a little bit too much, and so I was basically undergoing an allergic reaction. Um, Anyways, the hives eventually went away on its own. So we're going to learn more about the different kinds of white cells, and what they do and what it means if you have too much or too little of them. Okay, red cell first though. Right? Red blood cells or RBC. Right? The fancy name for RBC is erythrocytes. Erythrocytes. Right? So cytes means cells in Latin, and erythro is red. Right? So erythrocytes. They do not have a nucleus, right? Even though they are part of us and we're eukaryotic organism, they don't have a nucleus, and that allows them to have a biconcave shape, which increases the surface area. So what does biconcave means? Cave means like it's like indented, if you would, and biconcave means it's indented on both sides. So if you think about like a little oval shape here, and you're basically pinching in at the top and the bottom. We're looking at it from the side, right, like that. This is called biconcave, right? It's like a little pinched in a disc. And if you look at it from the top, you can see that's what the indentation is. And the purpose of that is basically to increase the surface area for gas exchange. Remember, the purpose of the red cells is to allow distribution of oxygen and actually carbon dioxide throughout the body. Right? Remember, you're not just delivering oxygen. You got to pick up the carbon dioxide and bring it back to the lungs and excrete them. So that would be the job of the red cell. The molecule, the proteins actually, that allows the red cells to transport oxygen and carbon dioxide is hemoglobin. Hemoglobin. You might remember learning about hemoglobin from the last semester. It's a protein with quaternary structure. It has four smaller peptide to it, and at the center of it, at the center of each of these smaller chains, is where you will find the heme group. Okay, the heme group contains a metal ion for transporting oxygen. What metal am I talking about? What metal do you have in the blood that allows you to transport oxygen? If you don't have enough of it, you might get anemia. That's right, iron. Okay, so the heme group contains iron. That's what the green little circle is. Okay, so it contains hemoglobin. Hemoglobin contains iron. Deficiency, as in not having enough, or defect, as in something's wrong with the structure, either in hemoglobin or red cell, will lead to anemia. Okay? Anemia. So there are different causes of anemia. You learned about sickle cell anemia. That's a defect in hemoglobin. Okay, and that causes anemia, shortness of breath, feeling tired all the time, not able to distribute oxygen effectively, right? If you don't have enough iron in your diet, then you can get iron deficiency anemia. Okay, there are different kinds of anemia. And at the root of all these different types of anemia is either you don't have enough hemoglobin or red cells, or the hemoglobins are defective because of mutations. Okay, so that's a sickle cell anemia. We talked about that already in the first semester. There's a mutation, there's a point mutation in the DNA that codes for the sixth amino acid in the chain. And as a result, it causes the cell to misfold. Okay, the hemoglobin misfold, which in turn causes the cell to have a different shape. And these cells are not very good at transporting oxygen. Now, it's interesting. People who have sickle cell anemia, it turns out they are immune to malaria. Okay? Malaria is something that's spread by a mosquito, right? Okay. It could be deadly in some cases. And it turns out if your cells are sickle shaped, then the malaria parasite cannot infect them and it's not going to make you sick, right? So this is a heat map that shows you places where malaria is very common And here is a heat map that shows you people with the big S, small s genotype. Okay. So let's take a look at what this means. If you are big S, then you have normal RBC. Okay. If you have little s, then you have sickle cell anemia, sickle cell. Okay. But if you're big S, small s, then you are kind of in between. Okay. You don't have the symptoms of sickle cell anemia. You're fine. Okay? And at the same time, it gives you protection from malaria. So that's why this kind of genotype is very popular in places where malaria is prevalent, okay, because it offers them protection. And evolution has selected that as an adaptive trait for people who live in those regions. Okay. One more slide on red cell, I think. Uh, one more, yeah, two more slides. Okay. Red cells are formed from red bone marrow stem cells. Okay. I didn't mention to you earlier, but there are red bone marrow and yellow bone marrow. Yellow bone marrow is mostly for storing fat. The red one are the active one that makes blood cells, including red cells. Okay. Hematopoiesis, right? When red blood cell level declines, the kidney is going to make a hormone called erythropoietin. You can call it EPO for short. And EPO is going to stimulate red cell production. Okay? So if you don't have enough red blood cell in the body, then you're not really distributing oxygen. Your kidney will sense that drop in oxygen, and then it's going to pump out this hormone called EPO. EPO goes back to the bone marrow and says, "Hey, we got to make more red cells. We're not distributing oxygen effectively. Let's do it." And then more red cells will come out. Okay? So EPO it's something that you make naturally, but you can also produce them artificially. And it's used for cancer patients who are undergoing chemotherapy or radiation therapy because these therapies often they target rapidly dividing cells, right? And as a side effect, they will dampen your bone marrow and cause your blood cell production to be reduced. And so to counteract that, you give them EPO. Right? You can also give EPO to people who are undergoing dialysis treatment. Dialysis is for people with kidney failures. So instead of the kidney filtering the blood, you have to take the blood out of the body and then filters it and then put it back into the person, right? But when you take blood out of the body and send it through all these tubings and things like that, a lot of times the red cells get destroyed, and then the person will have low red cell count. And so if you give them a shot of EPO, it's going to compensate for that. So that is red cell production and EPO. Red cells don't live forever in the body. Their lifespan is about 120 days, which works out to be approximately, 3 to 4 months. And when they are too old, they will be destroyed either at the liver or at the spleen. Okay. Your spleen is on the left side of the body, like around here at the side. Okay. And you only have one. Okay. And that, amongst other function, it is going to help break down red cells. So basically, they're going to be eaten up by this thing called macrophage, which is type of white cells that we will talk more about later on. What happens to the red cell when they are broken down? Well, iron is a scarce resource, so you really want to recover that, okay? And the protein as well. We don't want to waste them. So you break down the cellular portion and you extract the protein and then the iron so that you can use them to make new ones in the bone marrow. Whatever is not recovered, the remaining portion of it is going to be broken down into something called bilirubin. Okay? Bilirubin. Bilirubin is a yellowish substance, and that is what makes the urine and the feces the color they are. Okay, the yellowish color is the bilirubin. Now, not today, but next class, we will talk about a situation in babies where they experience a lot of red cells destruction, and their body is trying to break down all these hemoglobin that are coming out from the damaged red cells. And as a result, they have a build-up of bilirubin on their skin because their tiny body is not able to get rid of them fast enough, and it caused them to be a little bit yellowish. Um, Anybody know what condition I'm referring to? What do we call babies that are born a little bit yellowish? Yep, I have an answer coming in. Anyone else know what I'm talking about? Starts with, yep, that's right. It is jaundice. Okay, jaundice. So we're gonna talk more about that next class. For those of you who have not heard of it, okay, it's called jaundice. All right, and it's because of a buildup of bilirubin in the body for babies. Yeah, it's very common in newborn, especially for Asian Asian babies, apparently. Okay? I will share my story with you next class. Okay, that's it for red cells. Let's move on to white cells now. White cells are leukocytes. Leuko is white. Okay, cyte is cell. So red cell is erythrocytes. White cells, leukocytes. They are also produced in the bone marrow. Remember, all of your blood cells came from the same stem cell, okay? The same kind of stem cells, right? And the process again, it's called hematopoiesis. Where do we find white cells? They are not restricted to your blood vessels like the red cells. Red cells are only going to stay in the blood vessels unless there is a cut. Whereas white cells, they can be found in the blood, of course, but they can also be found in tissue fluid and some organs. There's a special ability for the white cells, which is shown here in this thing. They could squeeze themselves out of the capillaries. Remember, the capillaries are leaky by nature, right? So there are some gaps in the walls, and the white cells are able to, squeeze through those tiny gaps and go into the tissue. And in the tissue, they might start attacking things that are not supposed to be there. Okay. They will help with tissue maintenance and repairing in some cases, but primarily they defend against pathogens. Okay. Pathogens are organisms that cause you to become sick, like bacteria, viruses. What else can cause you sick? Has to be like a living thing, okay, that cause you sick. The cancer is not a pathogen, okay? If you have like cyanide poisoning, that's not a pathogen. It has to be like a biological thing, okay, a living thing. Right? So besides bacteria and viruses, what else can infect you? Mm-hmm. Parasites, good. One more. Starts with a F. That's right, fungus. Okay, fungus. Plural for fungus is fungi. Okay, so all these things are pathogens and they can go into the body and make you miserable. And the white cells are the defender of the body. They will help clear these things out as best as they can. They also help, do a little bit of housekeeping. Right? Any cells that are dead or if they are dying, they're not doing too well. The white cells will also destroy them. And remember, I told you the red cells, when they're too old, they will get eaten up by the white cells in the liver and the spleen. So that's part of the housekeeping and cleaning function of white cells. Okay, so I already told you red cells are confined to the blood, but white cells, they can come out. Okay? They could squeeze themselves out between the gaps and go, roam around in the tissue. All right, that's enough talking. Let's take a note here. This is what I'm gonna do. Your paper. I'm gonna have white blood cell here, and then I'm gonna have two categories, and under each category, I'm gonna have more categories. Okay, category, and then there's gonna be like more categories. Okay, I'm just telling you what to expect in terms of like how you space your notes. Right, but that's what it's gonna look like. So here we go. Let's start at the center. I probably should label this thing. It is types of WBC, white blood cells, okay? Leukocytes. So let's start with WBC. Now, just stop for a second. Don't write this down. Just listen, okay? In case you are a little bit confused, again, you don't have to write this down. I'm just reminding you what's going on, okay? Blood has plasma, and then it has formed elements, right? And then under formed elements, we have RBC, WBC, and then platelets, right? Okay, so now we're looking at WBC. So under WBC, there are like different ones. That's what we are writing right now for this note. Okay? So you don't really need this. I guess I'll put it on the side, okay? You don't have to draw it now. Look at it later. Okay, I'm just gonna put like a I don't think you can read that. Um There you go. Maybe it's a little bit better. Okay, so we're focusing on the white cells now. Okay. So here we go. Here we go. Here we go. WBC, white blood cell, and there are two categories of white cells. Now we can actually classify these cells using various categories. Okay, this is just one way of classifying them. Right? For example, right? Just to help you understand what's going on. If I give you a bunch of fruits and I ask you to categorize them, there are different ways to do it. Right? You can classify them uh, based on size. Okay. Okay, so the pineapples and the watermelon goes together, apples and lemon goes together, right? You can classify them based on whether you can eat the skin or not. So apples and pear goes together, pineapples and banana goes together, right? Like there are different ways you can categorize them depending on what you're trying to achieve, right? So here, this particular chart that I'm making for you is just one way of categorizing them, and it's based on what they look like. Okay, it's based on morphology. All right, so this is called granulocytes. Granulocyte. So a white cell is considered as a granulocyte if they contain granules. It contains enzyme. Sorry, just gonna say granules. Okay, so granules basically has enzymes in them. Enzymes or chemicals in them. Okay, these things do different things. Right? I'll tell you what they do later on in specific examples. But basically, they have granules, contains granules. Think of them as like the lysosome, okay? But it's like a special kind. Right? So you can see them under the microscope. You can see these granules, and we call them granulocytes. And right, you can probably guess it. Yeah, you either have the granules or you don't have the granules. And when you don't have the granules, it's the exact same word with an extra letter at the front. Agranulocytes. Agranulocytes. So these ones have no granules. Can be seen under the microscope. Okay. If you see granules, granulocytes. If you don't see granules, agranulocytes. It's just based on how they look. Okay. Of course, we can categorize white cells based on their functions, right? That's another way. But this is based on how they look. It's based on morphology. Morphology. Okay. Based on how they look. All right. Let's take a look at the different kinds of granulocytes. Number one, I guess. Number one right here. We have neutrophils. Neutrophils. Okay. Neutrophils are the most abundant white blood cell. Okay. And they are phagocytes. They are phagocytes. What are phagocytes? Phagocytes are cells. Cytes is cells. You should know that by now, right? Like leukocytes, erythrocytes. Phagocytes are cells that is capable of doing phagocytosis. You learned you learned about phagocytosis from me last semester, right? It's like a cell eating things, right? So essentially, what these neutrophils can do is they can go around and eat things up. If they see a bacteria, that's not supposed to be in the body. Let's go eat it. If they see a cell that's kind of, dying and not functioning properly, that's not supposed to be here. Let's eat it. That's basically neutrophils. Okay, it's approximately 70% of your white cells. Not 70% of your blood, 70% of your white cells. Okay? Remember, only 1% of your blood is white cells. Of that 1%, 70% of it is neutrophils. Okay? Next one is eosinophil. Eosinophils. Right? So eosinophils, right, they do several things. I'm just gonna keep it simple for you. So I'm just gonna emphasize one of their functions. Eosinophils, they are important for fighting parasites. Okay, by secreting chemicals. Okay, that's the main way of fighting a parasite. If you have like a parasitic infection and those parasites are really big, you can't phagocytize them. You cannot eat them. Yeah, so you have to secrete chemicals to attack them. And then there is basophil. Basophil. Okay. Basophils, they are circulating throughout the body and can trigger inflammation. Again, they do other things. If you read about this online and then you see that it has some other functions, like it's not wrong, okay? They sometimes have overlapping functions too. Yeah, but I'm just trying to highlight one of their functions, right? For the purpose of what we're learning. Okay. So inflammation, if you think about like an allergic reaction you have in the summer with the pollens and everything, okay? That's a kind of inflammation. There is another kind called the mast cells. They are related, right? Instead of circulating throughout the body, they are generally anchored to tissue and can also trigger inflammation. Okay. This is more localized though. Right? Think cutting a finger. You cut your finger, it's gonna be red, it's gonna be, pulsating, it's gonna hurt, it's gonna feel warm, might be some swelling to it. That's mast cells. Okay? This one is more like systemic type, bodily type of inflammation. So there you have it. These are granulocytes. They have various chemicals and enzymes in their granules. Okay, for example, okay, so like if you have an allergic reaction, right? Like the allergy pills that you take, what's in them? It has anti something. What's the something I'm looking for? Makes you drowsy. Hmm. Thanks, Kyle. That's right. It's antihistamine, right? So in the granules of these things, right, it contains histamine, right? So the granules basically has a mixture of enzymes and chemicals, and these chemicals allows these cells to, perform whatever function they perform. In the case of inflammation, they secrete histamine. Okay. On a side note, the basophil, for example, they also secrete something called heparin. Does anybody know what heparin does to your blood? Heparin. May have heard of it before. No. Okay. Heparin is a blood thinner. Okay? It prevents your blood from clotting inside your blood vessel, basically. Okay. So, they have a lot of chemicals, and all these chemicals do different things, and they're all stored in granules. That's what makes them granulocytes. Okay. Granulocytes. All right, so let's move on to agranulocytes. Agranulocyte. They do not have granules that are visible under the microscope. Can't see them. Um, and we have monocytes. Monocytes. Okay, monocytes. You can think of them as like inactive. I was at the library during the summer, and I found a comic book, I guess. Yeah, it's originally written in Japanese, but they translated to English. Um, I think it's called uh this. I forgot exactly what the name is. Okay, I'll show it to you in the break. But it's a comic about these cells in the body. Okay, and it's very accurate. So for example, the monocyte in the comic was depicted as these like freshmen coming into the police academy. Okay, like they don't know what's going on, right? They're just like, trying to train up and be strong so that they can fight. Okay. And so, they're inactive. They're not experienced. Okay. They don't really know what's going on. But once they are trained up, then they could become the macrophage. Okay? Macrophage. So these are like the seasoned killer, if you would. Okay. They're the seasoned police, and they are able to fight off the infection. Right. So this is active. So this is like what we call a differentiation. It's a one-way transformation that allows the cell to become more capable of clearing out an infection. Okay, so sometimes you will have macrophages that stay at a particular organ because they are regularly needed there. So you would have it at the lungs, for example, because we constantly breathe in stuff from the air that might be harmful, and you want to clear them out as soon as they enter. You will have macrophages in the spleen, right, in the liver to clear out worn out red cells. Right? So they came from monocytes. Okay. We also have lymphocytes, lymphocytes. And there are different kinds of lymphocytes. I'm just going to list them here for you, and we're going to learn more about them later. There are T cells, B cells, and NK cells. Okay? We will learn a lot more about them in lecture 3, but briefly, uh, T cells, they coordinate immune response. B cells, they make antibodies. And NK cells, NK stands for natural killer cells, okay? That's actually what they're called, natural killer cells. And amongst other things, they kill infected cells, right? And cancer cells. So there you have it. Okay. Give you a moment to copy. All right, I found the name of the comic. You can find a copy at your local library if you're interested. It's actually very medically accurate in terms of what these cells does, um, and it turns into a comic, which is entertaining. For me, it was. Anyways, it was just three volumes of it. Okay, why don't we take a break and we'll do more when we return. Let's come back at 1:45. Okay. Sorry, 9:45. 9:45. Okay. See you all in a bit. Hello, everyone. Let's continue with our lesson. So that's everything you should know for your white cells. Reorganize the chart, make cue cards, whatever you need to do to remember them. Right? So that's what they look like under the microscope. We have the granulocytes here, and they actually have multiple nuclei in some cases. The neutrophil here, you see a distinct 3 lobes, and then for the eosinophil, you can see is just 2 nuclei here. You're not expected to identify them, but I'm just showing you what they look like under the microscope. Okay. Moving on. The last one is platelets. Platelets are also called thrombocytes. Thrombocytes. So we have the erythrocytes, which are the red. We have the leukocytes, which are the white. And then now we have the thrombocytes. Okay. They are not actually cells. Instead, they are cell fragments that are broken off from a much larger cell. In this picture, you can see that larger cell that is called the megakaryocyte. Okay, megakaryocyte, and bits and pieces of platelets just kind of breaks off from that. You produce about 200 billion platelets per day. They don't have nucleus and they don't last very long. Okay? They only last about 10 days unless you use them sooner. Okay? If you have a cut, then you got to use them. Right? Otherwise, they will stay in your body for about 10 days. And the process of clotting your blood, right, coagulation, right? We learned earlier, but it's also called hemostasis. Don't confuse hemostasis with homeostasis. Homeostasis is maintaining internal balance in general. Hemostasis is maintaining balance in the blood, right? So like basically stopping the bleeding so you don't bleed out. And that is the main function of platelets. Okay. So let's take a look at how coagulation occurs. Um, and we're going to take a short note on that. This is really short. Right? This is really short. Okay, so coagulation. There we go. So you might remember I told you in the first lesson, blood does not like rough surfaces, right? As soon as blood passes through a rough surface, then they tend to have a blood clot. And so let's say there's an injury, right? There's some kind of thumbtack going into your blood vessel or something, and that damage actually exposes some of the underlying tissue in your blood vessels. Okay, so here, damage to blood vessel exposes underlying tissue. And that releases something called thrombo thromboplastin. Okay. Damaged tissue releases thromboplastin. That's the first step. Okay, so if you don't have a cut right now, you will not have thromboplastin in your system. Okay, so these thromboplastin comes out basically. Now, earlier today, I told you in your plasma, you have various kinds of proteins, and some plays a role in coagulation, right? And I don't know if you remember, one of them is prothrombin. Prothrombin. When you see the prefix pro, it means it is inactive. Inactive. So you have prothrombin in your blood right now, and they will be switched off, which makes sense because you're not bleeding. You don't need to clot your blood. We don't want the blood to clot in the blood vessels like randomly. That's dangerous, of course. So we keep them off. So how do you turn it on? That's what the thromboplastins are for. When you damage the tissue, when you damage the blood vessels, the damage releases the thromboplastin, which will then turn on the prothrombin. Okay, you need a little bit of calcium to do this as well, and it basically cuts the protein and it turns into the active form, and it's called thrombin. Thrombin. This is active now. Any questions so far? Are you following? Please let me know because I'm gonna tell you more after this. Okay. Also in your plasma is an other coagulation protein called fibrinogen. Fibrinogen. If you see the suffix ogen, it can also mean inactive. And again, if you're not bleeding right now, your fibrinogen are all switched off. They're not activated. They only get activated if there are thrombin in your system. Okay, so you also need a bit of calcium, and that turns it into fibrin. Fibrin. Fibrin are basically these long strand of white fibers, okay, and it forms a net. Okay? So the net is going to trap the platelets, and it's going to trap some red cell as well, and it basically help seal the wound, and eventually it becomes the scab. After your wound heal, there's a scab, right? And people like to pick at it, and then it bleeds all over again. But that scab is basically made up of fibrin and platelets and trapped red cells, and it basically seal the wound and prevents bleeding to continue, right? So this is the coagulation, right? So you have these things, prothrombin and fibrinogen in your plasma. They're circulating throughout the body. They will only get turned on one at a time if there are thromboplastin. And when do you get thromboplastin? Only if you damage your blood vessels. Okay, so it's a cascade of events, like a domino. Push the first block and then one thing leads to the next. Is this okay? So here in this picture, you can see these white strands. Those are the fibrin. That's what you're trying to make when you have a cut. And then this will trap. You can see the red cells being trapped, and then can't really see the platelets in this picture, but they will come. They will come. And that's all for the first half. Can you tell one's 1, 2, 3 step? So do you mean do you want me to repeat 1, 2, 3? Is that from the notes? Okay. If the blood vessels are undamaged, then we will not have thromboplastin, and the blood will just pass through no problem. Okay. As soon as you damage the blood vessels because of a cut or something like that, then the damaged tissue will release the thromboplastin. Okay, so that's the first block in the dominoes, right? And the thromboplastin, once is released, okay, it will cause the prothrombin to get turned on, which in turns will turn on fibrinogen, and then it becomes fibrin. Now, it's not going to happen throughout the entire body because the thromboplasts are only released at the site of the damage, right? So this fibrin net will only form right here, and it will seal the wound at that particular location. Okay. Elsewhere, blood is not going to clot, and we don't want it to, because there's no bleeding, and we don't want blood clot to spontaneously form on its own. Is that better, or like do you have a specific follow-up question, Sheila? All right. So that is it. Right? This is a summary of everything we talked about. You don't have to memorize the numbers. Right? Red cell, transport carbon dioxide, oxygen, you know, eosinophil, kill parasitic worm. Right? It's basically what we wrote down, right, in a summary uh, over here. Right? We have the plasma portion, 55%, cellular portion. Right? So you can read this and have a better understanding of it. Okay. We have not done any polls for the whole day. Let's do some. All right. So the correct answer is basophil. A basophil is a granulocyte. We wrote it down, right? If you have trouble memorizing these things, then maybe put it on your help sheet for your test. It's fine if you need to do it. How about this one? If you have a worm, who's you gonna call to help get rid of it? They are too big to be eaten up by cells. What do you need? Very good. Most of you got it right. And again, you just have to look at your notes, right? It's not rocket science. Uh, Worm is this one, parasite. Right? So if the person has a lot of eosinophil, they're probably fighting some kind of parasitic infection. The correct answer is C. Okay, C. Inflammation is basophil and mast cells. Okay, allergy is also basophil. Internal bleeding has nothing to do with white cells. All right, another one. It's the last one. Yep, very good. Hemoglobin is found in red blood cells. Red blood cells. Excellent. That concludes the first half of the lesson. Now we're going to do the second half. Second half is genetics of blood type. Okay, so we gotta go through the genetics, and then we're gonna go through the transfusion, and then we're gonna solve problems with them. Uh, do you remember much from last semester? The genetic stuff, like if I say allele, phenotype, genotype, do you know what I'm talking about? If you want me to go through them again, just raise your hand or something.
Yeah, you can refresh, please.
Yeah, do a quick review. Yeah, yeah,
Yes.
sure, no problem. Okay, alleles are different version of a gene. Okay, different version of a gene. So big A, small a, right? Big B, small b. Phenotype is the um, manifestation of the genes, is the expressed um, is the outcome, if you would, is the appearance or outcome of the gene. So like if you are big B, small b, then you have blue eyes. Okay, so the blue eyes is the phenotype. Okay, or brown eyes, that's the phenotype. Whereas the genotype is the genetic makeup. Genetic makeup. Are you big B, big B, big B, small b, small b, small b? That's the genotype. Right? Remember, two people with the same genotype might have the same phenotype. Okay, so that's the background. All right, we're gonna look at the blood type and the blood group, and actually, does anybody know their blood type? You can share it. You can make it up. I wouldn't know. I would never know. Okay, but like, do you know what your blood type is? Type it in the chat box if you do. Unmute and tell and enlighten us. Yeah, yeah. So like, good. I got a few coming in. People are saying A, B positive, O positive, right? So what you are telling me are actually two blood groups. There's the ABO blood group. That's what the A, B, and then the O is representing. And then the plus that you're talking about, that's an other blood group. It's called the RH blood group. But there are many other different things that are on the surface of the cell. Um, and if you are going to do like a planned transfusion, they often will match as many of these blood group as possible. to make the transfusion as safe as possible. So there's the ABO, there's the RH, that's the positive and negative you guys are talking about. But there are other ones like Cal, Duffy, okay? Some of them are really weird name, LW, MMS. All these are extra things that are on the surface of the red cells and they contribute to other types of blood group. So blood type refers to the inherited antigens present on the surface of your red blood cells. Okay. What are antigens? Antigens are basically things that are found on the surface of the red cell, on the surface of a cell in general, but in the context of what we are talking about, we are interested in the things that are found on the red cell. These things include membrane proteins, carbohydrates, glycoproteins, and glycolipids. Okay, again, you might remember this last semester. The glycoproteins and the glycolipids, they are responsible for cell-cell recognition, right? Okay. And it's because they form part of these antigens that are on the surface of our cells. There are over 40 blood groups that has been identified. If I click on the link, it's going to take you to everybody's favorite online source, Wikipedia. You can see there's ABO blood group. There's MNS, RH, Lutheran, Cal, Lewis. Right? All these are blood groups. They are all antigens that are found on the surface of the red cells. Okay. Um, the most important blood group is ABO and RH though. Okay. The other ones, there are differences within the populations, but typically they play a lesser role when it comes to transfusion. But they check them as well. Okay. They do check the compatibility for some of those groups in a planned situations. But if it's an emergency, then ABO and RH is the most important blood group that they look at in terms of compatibility. I see a lot of AB positive. Well, not a lot. Okay, look, but I see two two AB positive. That actually is not very common. Okay, the most common blood type according to Canadian Surface in Canada is O positive. Okay, a lot of people are O positive, followed by A positive. Okay, AB negative is really rare. All right. So, what determines the whether you're A or B or A, B or O? It has to do with the presence or absence of antigen A and antigen B on the surface of the red cell. And these antigens are basically proteins, okay? And if it's a protein, then you would have a gene for them. Right? So there are three alleles that are responsible for the ABO blood group. We have IA, IB, and little I. So let's take a quick note on blood type here. Lots of notes today. Okay. Genetics of ABO blood group. We'll do RH next class. We don't have time today. So three alleles in the population. Okay, this is the way they write it. Okay, don't ask me why. IA, IB, or little I. Now I want to ask you, even though there are three alleles in the population, human population, how many different ones can you have in your genome at most? How many alleles can you have? for ABO blood group in your genome at most? Two is correct because we are diploid, right? We have two sets of chromosome. You will inherit one allele from mom and one allele from dad. So even though there are three alleles, at most you can have two different ones. So how many combinations can we generate? Well, let's check this out, okay? If you get the IA, then you can make antigen A. Antigen A. If you have IB, then you can make antigen B. If you are little I, then you do not make anything. So type A blood. This is the phenotype, okay? Phenotype type A. How are you type A? Well, if you're type A, then your red cells will have A on them. Okay, A protein on them. This is the letter A. Okay, and how do you put A on your red cells? Well, you will need the IA allele. So you can either be IA or you could be IA little I. Right? Doesn't matter if you have two copies or one copies of the IA, as long as you make IA, as long as you make antigen A, then you are type A. Okay, remember, little I doesn't make anything. So in this case, it's recessive compared to IA. Okay, think of them as cookie cutters. If I give you a star cookie cutter, you can make star cookies. It doesn't matter if I give you one or I give you two, you can still make them, right? So that's what it is. Doesn't matter if you have two copies or one copy of the IA, you're still going to make A, and then that's going to make you type A. So two possibilities for someone to be type A. What about type B? And what does it mean for someone to be type B? If they are type B, they will have B antigen on the surface of the red cells. What are the possible genotypes then? And remember, these are genotype. This is phenotype. Okay, help me out here, guys. What's the genetic makeup of someone who has type B blood? Am I confusing you? Oh, it's just taking some time to type. Okay. Thank you. Oh, with the shift 7 too. Very nice. Okay. This is IB or IB little I, right? IB little I. Very good. And so you can, now you need the moon cookie cutters. Then you can make moon cookies, right? Again, it doesn't matter if you have just one cutter or two cutters. You make them, then it's fine. Okay, what about type AB? Type AB, right? For someone who's type AB, they would need to have A and B on the surface of their red cell. And what's the only way to achieve that? What genotype do you need to have type AB blood? That's correct. You need IA and IB. There are no other ways. Okay, you need the moon cutters and you need the star cutters to make both kinds of cookies. And then type O. Type O means nothing. Right? No A, no B. It's just type O. So how do you have nothing? Little I, little I. That's all. So the ABO blood group And we're not talking about the positive or negative yet. That's next class. For the ABO blood group, there are three alleles, six genotypes, one, two, three, four, five, six, and four phenotypes. Okay. Genotypes, six combination, six types, phenotype, four types for the ABO blood group. Any questions? If you don't understand this, you gotta ask me, right? The rest of the class is gonna be built upon this. If you're lost now, you need to clear things up. Otherwise, you'll be continue to be lost for the rest of the class. Ask me anything. If you have a question, someone else probably has the same question. Okay, I will take your word for it. All right. Hopefully, it's not too difficult. Let's do a genetic problem right here. Okay, I'm gonna copy it to the OneNote here so we can do it together. Gotta make it smaller though. There we go. A man with AB blood is married to a woman with AB blood. What blood types will their children be in what proportion? All right, so let's take a look at the man here. The man has AB blood. As we just learned, if a person has AB blood, there's only one possible um, genotype, right? So the man is IAIB. Okay, and he's gonna have some kids with the woman who's also AB blood. So she's also IAIB. And then we're gonna cross them. Gonna cross them. Remember, the alleles will separate during formation of gametes. And so half the sperm will have IA, the other half will have IB, and we're going to be doing a Punnett square for it. Let me just restart the program. It's not letting me draw straight lines. Okay, so you remember Punnett square, I hope. Doesn't matter where you put the men and where you put the woman. It's not gonna change the result. So the men, like I said, 50% of the sperm will carry the IA allele, the other half will carry the IB. For the woman, half the eggs will carry the IA, the other half will carry the IB. Okay, just gonna put an arrow here to show you. Again, doesn't matter where you put the men or the woman. And so if this sperm fertilizes that egg, the kid will be IAIA. If this sperm fertilizes this egg, then the child would be IAIB. And similarly, you can continue to fill in the rest of the table. The Punnett square, right? That's what it's called. And so let's interpret the result. There will be a 25% chance, 1 out of 4, that their kid will have type A blood. 50% chance type AB, and 25% chance they will be type B. Is that okay? All right. Next example. Just a quick story. One of one of the fears my wife had when we had our first child was that she was worried we're gonna bring home the wrong baby. Right? So she gave me a mission. She's like, when the baby's born, you stay with the baby at all times. Wherever the baby go, you go. Right? And I was like, but what about you? Like, don't you need someone to like watch you? She's like, don't worry about me. Okay, I'll be fine. Just watch the baby. Right? Because her sister gave birth in Hong Kong, and in Hong Kong, um, the baby is not always with the parents after they're born. Okay, so they might have read some stories about bringing home the wrong baby, and she was so concerned. But it turns out in Canada, the baby stays with the mom at all times. So I'm pretty sure I brought home the right baby. So here in this question, there's a mixed up of babies at the hospital, and we're trying to figure out which baby belongs to which parent. Okay. Now this is just for fun, okay? Like if there is an actual mix-up at the hospital and they realize it, they are not going to be using blood type to figure out what babies are, who the parents are. Okay. What technique do we use if we try to figure out the identity of the babies? That's right, DNA fingerprinting. We learned that last semester, right? Okay, so that's what they're going to do. Okay. But this is just like an intellectual exercise. All right. So we have two babies here. Baby one is type O, baby two is type A, and we got the Jameses here and then the Marshalls. You do not need to do a Punnett square to figure this out. You can just, you know, reason through it. So I'm gonna give you a moment to try that. Okay. Think about who's who. And you can let me know when you have an answer. All right, I have some answers coming in. Feel free to message me, private message me if you prefer that. But let me know what you think. All right. Thank you for all the answers. Let's take a closer look at how you would approach a problem like this on a test. So baby one is type O, and there is only one way to be type O. That is little I, little I. Right? For someone to have type O blood, they must, they must inherit a little I from each parent. Okay? Two little I's, one from mom, one from dad. Let's take a look at the Jameses here. Okay? Type B, I mean, like he could be IB, or IB little I. We're not really sure. But the husband, type AB, is IB. Right? So based on this alone, you can say not possible for him to have a type O baby. It doesn't matter who he marries. Doesn't matter his spouse, blood type. It's impossible for an AB person to have a type O baby because of what I said. To produce a type O baby, you need to contribute one little eye. He does not have a little eye to give. For that reason, baby two is theirs. If there are only two babies up for grabs, right? It's one or two, then it's definitely not number one. It has to be number two. Does that make sense? AB individuals cannot ever have a biologically baby. That's type O. And you can adopt one if you really like type O babies, okay? But you can't have one of your own. Doesn't matter who you marry. Now, some of you might be confused, but how is it possible for two type B individuals to have a type O baby? Well, remember, there are two ways to be type B. You can be IB, or you can be IB little I. So in this case, both of them must be IB little I. And if you do the Punnett square, there's a 25% chance, 25% chance they are able to have a type O child. Right? If anything, they cannot have a type A baby. Right? They don't even have the IA allele, right? So that's how you do these kind of questions. You just kind of logic your way through it. It gets more complicated once we include the RH, but we'll do some examples of that sort next class. All right. I hope you are doing just fine. And like I said, right, I always encourage you guys to ask questions. All right, here we have the Shrek family. We got the king, we got the queen, we have Fiona here, and then there are kids. Please figure out the blood type for Fiona. Okay. I will give you, I don't know, 3 minutes maybe. Okay. If you have the answer, let me know. Okay. We are looking for the blood type of Fiona. Okay. So 3 minutes. All right, that was 3 minutes. I have a couple answers coming in, and they're uh, all over the place. Let's take it up and see how we would do a question like this. First of all, you would start by writing down the genotype of people you are certain about. And there are only two blood types that you know for certain, which is AB and O. So the king here has type AB blood. He must be IAIB. For Fargo here, he is type O, so he must be little I, little I. Okay, little I, little I. That's all we know for certain. Everyone else in the pedigree, we only know half of the genotype for certain. So let's write those down. The queen would be IA something. We don't know if she's IAIA or IA little I. Okay, we got to figure that out. Shrek here is IB something. And Fergus is IA something. And Felicia, she's IB something. Okay, so that's all we know for now, for certain. Right, everything else we gotta use some reasoning and logic to figure out. So let's start with Fargo here and try to understand where he got the little eyes. Um, as I told you in the last example, if someone is type O, they have to get a little eye from each of the parents. So one of the little eyes here is going to come from Shrek. The other one is going to be from Fiona. Okay. Now, where did Fiona get her little eye? Well, it's not from the king because the king can only contribute IA or IB. It must be from the queen. All right, so you can track the little eye throughout the generations, right? Okay, let's take a look at Fergus here. Fergus is IA something. Where did he get the IA? Well, it's not from Shrek. Shrek is type B. He cannot pass on IA. He doesn't have one. So the IA must have came from Fiona. Okay, so Fiona is IA little I, type A. That's the only possibility. Okay, so let's wrap this up. The oops, sorry. What about the rest of the genotype? Well, the rest of it, it must be from Shrek, and it wouldn't be IB because if it was, then the blood type would have been AB, right? Therefore, it must be little I. For Felicia, the IB came from Shrek. Fiona doesn't have a B. The other one, it's going to be from Fiona, but it's not going to be the A because otherwise the blood type would have been AB. Therefore, it must be little I. So there you have it. That's the solution. Any questions? Fantastic. Let's do another one here. This is a poll question. Poll question. All right, let's take it up. We are looking for John right here. For John. So let's look at his kids. Sue here is little I, little I. So one of the little I must have came from John, right? Yeah, one of the little I must have came from John. Another daughter here, Mary. Mary is AB, so IA and IB. Right? Uh, the A is going to be coming from, I mean, like, we don't know if the A come from the mom or the dad. However, the B has to come from John, right? The B cannot come from the mom because the mom is type A. Type A person do not have an IB to pass on. So John must be IB little I. And we know the A therefore came from Harriet, right? So that's the answer. Right? It has to be IB little I. There are no other possibility that would account for these results. Okay, one more. We are looking for Mona. Mona. Okay, anyone else? It's been almost been 2 minutes. Should I take it up? Okay, let's take it up. So same family tree as the last one. It's the exact same one, okay? So we already figured out John is IB little I. We know Harriet is IA little I from the previous question. And so where did Harriet get her little I? Well, her father, Ed, is type O, so Ed must be little I, and therefore this little I came from Ed for sure, because that's the only thing Ed can contribute, right? And therefore the IA is going to come from Mona. But that's all we know about Mona, right? We don't know what the other half is, right? The other half could be, a B, it could be another A, it could be a small I. We just don't know. It's not necessarily A, B for sure, right? So none of these options actually works. So we need more information. Okay, she can be IA little IAIB. We just don't know. The only thing we know for certain is she's definitely not type O. Okay, but beyond that, we don't know. The answer is E. Questions? Okay. All right. All right. Next, we're gonna do antigens and antibodies, and we're gonna do transfusion, and then we'll be done. Okay, so we're almost there. Lots of information from today's lesson. I know, I know. Okay, remember about the videos I told you about. I I have to repeat a lot because people sometimes they don't listen. Go to the lab folder, go to the lab videos. There is blood typing explained. Okay? Watch that after class. Even if you understand what I'm telling you, watch it. Okay? It's gonna help consolidate the concept. Okay, so here we're gonna go antigen, antibodies, and blood transfusion. And blood transfusion. So let's make a table here. Actually, you know what? I'm just gonna take it from the study guide. it is, assuming you have it, uh, more pedigree in the study guide if you want. Where is the table that I'm looking for? Oh, right here, number 6. Okay, number 6. That's the table I'm gonna fill in for you now. If you have it print out, excellent. If you don't have it, then make your own. It's not too difficult to make. There we go. All right. But before we do that, let's recap some of the uh, key concepts here. Oops, slowly off to the one side. Let's bring it back over here. All right, let's start with antigens. What are antigens? Antigens are basically molecules on the surface of RBC, okay, as determined by your genes. Right? We talk about antigen A, we talk about antigen B. That's the ABO blood group, but keep in mind there are other ones which we are not focusing on. That's antigens, okay? And also, I should mention, antigens are not just on red cells, they are on all your cells. But when it comes to blood type, we're only interested in the ones that are found on your red cell. Okay? Antibodies. What are antibodies? Antibodies are Y-shaped proteins that target foreign antigens. Your body is able to recognize antigens that are from outside of your body. Right? If they're from outside, like for example, like a bacteria coming in, it would have antigens that you don't have on your cells. And to fight them off, you would produce antibodies for them. As a general rule, to understand this, you make antibodies for antigens that you don't have. Okay? Don't think of antigens as good or bad. It's not like that. Okay? Antigens are just stuff on the surface of the cell, and your antigens are going to be different from my antigens, right? Which is why you can't just like donate your organs to anybody, right? The person is going to reject it if the antigens don't match. Okay, so everybody has antigens on the surface of the cells, and you will make antibodies that would target and attack foreign antigens, the ones that you don't have. Keep that in mind. So here is our table. Okay. Type A blood. Type A blood, the genotype, as I have been telling you all day long, is either IA homozygous or IA little I. Well, what does that mean? It means on the surface of your red cell, on the surface of the red cell, you will have the A antigen. Oh, sorry, I gotta restart again. This thing is glitchy. It doesn't let me change color after a while. And I do like my colors. All right, here we go. Let's see if it works now. So on the surface of your red cell, which is what this thing is, you will have the A antigen. Okay, I'll use red for the A antigen. What about antibodies in the plasma? What kind of antibodies will you make? Well, yeah, exactly. Right? If you are A, then you do not have B in your system, then B would be foreign to you, and so you would have antibodies. They are Y-shaped. They literally are. That would target B. So we call this anti-B antibodies. Okay, anti-B antibodies. In other words, they attack anything that has a B on it. So for that reason, if you have type A blood, you cannot receive type B blood. Your antibodies will attack and reject them. More on that later. I'll fill in the last column later. All right, let's take a look at type B blood. If a person is type B blood, then they are either homozygous or heterozygous. And if you draw the red cell, I have to make this bigger. Okay, if you draw the red cell, what do they have on the surface? Well, the IB allele gene will allow you to have B antigen on the surface. And in this case, what kind of antibodies will you make? Well, I hope you know the answer. It is anti-A. You make antibodies to attack things that you don't have. Right? If chickenpox infects you, then you make anti-chickenpox antibodies. So for someone who's type B blood, they cannot receive type A blood. The antibodies will attack and reject the blood. Okay, so far it's fairly straightforward, I hope. Next one, for someone who's type O, there's only one way, which is little I, little I. And what do we draw on the surface of the red cell? What should I put on top? Nothing. That's right. Nothing. Type O is no A, no B, nothing. And since you have nothing on the surface of the red cell, you're going to make two kinds of antibodies. Anti-A, anti-B. Okay, because both of these would be foreign to you. And if you're type O, you cannot receive blood from A or B. Your antibodies will attack them. How about the last column here? Type AB blood. That's IAIB. That's the only genotype. And what kind of red cells will you have? What's on the surface? Since you have both alleles, you're going to have A and you're going to have B. And what about antibodies? What kind of antibodies will you make? That's right, nothing, none. You cannot make A or B because if you make anti-A or anti-B, you would end up attacking your own cells, right? Remember, the antibodies will attack the same letter. Yeah, so there are no antibodies. Now I gotta emphasize, okay? When I say no antibodies, I'm talking about the ABO only. It doesn't mean you cannot make antibodies for chickenpox. Okay? Like we're just talking about within the ABO blood group. Are we together, guys? You have to understand this before we talk about transfusion. I know I ask you every 5 minutes if you're okay, but I mean it. If you don't understand, you need to let me know. It's only going to get more confusing. Okay, okay, very good. Sorry, the my kid's school is calling me. I have to pick up. Okay, I'll be right back. Sorry about that. Thank you for waiting. Uh, let's keep going. Blood transfusion. Okay, blood transfusion. Let's take a look at the situation here. Okay. Um, Remember, I told you at the beginning of the class, blood transfusion that we are looking at is red blood cell transfusion only. Okay, red blood cell transfusion. The rules for plasma transfusions are different. In the past, I teach both, and then people get really, really confused. Okay, so like I'm just gonna stick with RBC transfusion here. Okay, so you take the blood out, you separate it into plasma and red cells, and we're only looking at the red cells portion. So there are three questions that you need to ask yourself when you are trying to figure out whether a transfusion is safe or not. So number one, first question, you need to know the antigen or antigens on the donor's cell, okay? On the donor's RBC. Number two, we need to know the antibodies in the recipient's plasma. Okay, what kind of antibodies does the person receiving the blood has, basically? And number three, will the antibodies attack the RBC? Okay, these are the three questions you have to ask yourself um, to determine whether the transfusion is compatible. So let's do an example. Let's do an example. Can A blood be given to a B person? Okay. I already told you no, right? But let's try to understand why. Let's try to understand why. First question, antigens on the donor's RBC. Right? So for the donor, because they are type A, you will have A antigens on them. For the recipient is type B blood. So what kind of antibodies will a type B person produce? You can think about it. If you need help, you can look at the chart. But remember, you make antibodies against things that you don't have, right? So what's going to happen if you go ahead with the transfusion? Well, the antibody is going to attack the red cells. Okay, this it's going to attack it as long as the letters are the same. It's going to attack it. Right? This is called a cross reaction. Okay, and cross reaction will lead to clumping, which is called agglutination. I know a lot of words. I'm so sorry. And then it will lead to hemolysis, which is RBC bursting. Okay, so that's not good. Right? The reason why a person needs a transfusion is because they don't have enough red cells. If you're gonna give them incompatible red cells and they all end up clumping together and bursting all over the place, that could be doing more damage than than not having a transfusion. Okay, and a person could die from something like that. Okay, how about this one? Can you give AB blood to a type B person? Okay, I'm gonna launch a poll. If you think this is safe, please choose A. If you think it's not safe, please choose B. Okay. Anyone else before I move on? No? No more answers coming in? Okay. Well, it's kind of half-half. More of you think is not safe than is safe, but it's like roughly half-half. All right, let's talk about this. Okay, A, B. Let's see. Question one. Antigen or antigens on the donor's RBC. This is the donor, the person who's giving the blood. They will have A and B on it. The recipient is type B blood. So just like the previous example, they will have anti-A antibodies. So if you go ahead with a transfusion, the antibody will attack anything that has A on it, and since the incoming cells has A on it, they will attack it. This is not safe. Not safe. All right. You might be thinking, but there's B, right? And then I'm B. Isn't that okay? It doesn't matter. If you see A, if you see something that the antibody recognizes, it will still attack it. Okay? So this is not safe. One more. One more. Let's do O to A. Safe for A, not safe for B. Much better this time. Okay, just a couple more seconds in case you wanna. All right. Everyone who answered got it right. Okay, I usually joke about um, about this thing. Right? If you don't understand the blood transfusion by the end of the course, please let me know which hospital you're working at eventually, and then I will try not to go there. Because if you give the wrong kind of blood transfusion, uh, it could be life threatening. Okay, so this is safe though. This is safe. Right? If someone is type O, there is nothing on the surface of the cell. Someone who is type A blood, they will have anti-B. And if you go ahead with transfusion, that's okay because there isn't any B on the surface of the red cell, and then the antibodies are not gonna, be attacking anything. So this is safe. If you think about it, O can be given to anyone within the ABO blood group. You can give it to type A, you can give it to type B, you can give it to type AB. Right? What's the name people refer type O to as? What do we call them? What kind of donors do we call them? That's right, call them universal donor. But as you will see next class, it's not quite right. Just because you are type O doesn't necessarily mean you can give it to literally anyone. Okay? Because there are other blood groups that exist, and we have to take those into consideration as well. Okay, so don't get too hung up on that term. All right, I hope I've done an adequate job explaining this to you, but for some reason, if you are not fully getting it, obviously we watched the lesson, watch the video recording that I was telling you about in the like the demo right here, okay? And I have a cheat sheet for you. Cheat cheat sheet. Okay. You can call it whatever you want if you don't like this name. But put it on a sticky tag, stick it next to your monitor before you do the test. This should help you out, okay? This tells you who can give to who. Okay, so obviously you can always give to the same type. If you're type O, you can give to type O. If you're AB, you can give to AB. All right. However, you can give to other people as well. Type O, you just follow the arrow. Type O, you can give to A, you can give to AB, you can give to B. Who can B give to? B can give to other Bs, or they could give to AB. Just follow the arrow, right? What if you are AB? Who can you give red cells to if you are type AB?
Only AB.
Only AB, that's right. Okay, so AB people can only donate to AB, but they can receive from everyone. Okay, for O, they could give it to everyone, but they can only receive from O. Okay. So some people say O is the generous one and AB are the selfish one, but that's actually not true because, and you don't have to know this, but I just want to tell you, the rules for plasma transfusion is completely opposite. Okay, it turns out if you have AB plasma, you can donate to anyone. And if you have O plasma, then you can only donate to the other O's. Okay, so like I said, there are different rules, right? But we are only looking at RBC transfusion. Okay, let's finish the lesson. If you are type A, who can you donate to? You can donate to other A's and follow the chart, the cheat sheet. You can donate to AB. Okay. But like obviously think about it, okay? When you have time to go through the lesson, think about it. Don't just memorize the chart. The chart is a handy guide, but I really want you to understand this stuff. If you're O, fantastic. You can give it to everybody. You can give the red cells to everybody. If you're AB, then you can only donate to other ABs. And that's it for today. All right. Study hard for the quiz. I'm not gonna. This is a very different platform, right? I'm not gonna be able to post the like the link for the quiz. So when the time comes, which is next Monday, you just have to go into the quiz folder and then you will just start the quiz. You don't have access to it right now because it's not within the time frame yet. Okay, it's going to be available on Monday between 6 a.m. to 11:59 p.m. And there will be like, 22 multiple choice and four multi-select questions. And you have to have the lockdown browser. Okay. I don't remember if I mentioned this already, but this is a little bit different from last semester. Okay. Last semester, the end point was 11:59, right? And I literally had people doing the quiz at 11:50 and they will still get the full time. That's not the case for this system. If it's due at 11:59 and you start at 11:50, you will only have 9 minutes to do the quiz. Okay? So don't wait until the last minute. That's what I'm saying. Okay? Just do it as early as you can, maybe between my class and your afternoon class, and then be done with it. Okay, I'll try to end a little bit earlier next class if possible to give you some time to do the quiz. Okay, thank you. See you guys. Have a good week.
Thank you, sir.